This is the fifth lecture on the nervous system. The lacrimal glands are located in the upper lateral portion of the eyelid and secrete tears. The tears are secreted through the lacrimal duct and open into the lateral surface of the upper eyelid. Tears move from the lateral surface to the medial surface and drain through the nasolacrimal duct into the nasal cavity. Tears contain lysozyme to help kill bacteria, as well as mechanically flushing out the eye. When we have more secretions than the duct can carry, the tears will spill over onto our face. This is also why our nose tends to run when we cry, because of the excess tears in the nasal cavity before they can evaporate. The conjunctiva is the lining covering the inside of the eyelids and across the anterior surface of the eyeball. When it is inflamed, it is conjunctivitis. An infected conjunctiva is pink eye. The eyeball itself has 3 layers called tunics. There is a fibrous layer, a vascular layer, and a nervous layer. The cornea is part of the fibrous layer. It is the transparent coat over the colored part of the eye, or iris. This is the part of the eye involved in cataracts. If you look at a person's eyeball from the side, the clear bubble you can see on the outside is the cornea. The sclera is the white of the eye and is also part of the fibrous layer. It is made of dense connective tissue and helps to give the eyeball shape. The retina is the nerve layer. It is the inner coat lining the posterior surface of the eye and is the actual sensory organ of the eye. The choroid is the vascular layer. It is a thin membrane with blood vessels lining the internal surface of the sclera. The retina sits on top of the choroid. This is why you can sometimes see little red blood vessels in the whites of the eyes, and also makes the blood vessels that are seen when looking through an ophthalmoscope on an eye exam. The lens is the transparent part of the eye that focuses the light on the retina. It is located behind the cornea and iris of the eye. The iris is the colored portion of the eye and is made up of circular and radial muscles that are arranged to allow and adjust how much light enters the eye by controlling the hole in the center called the pupil. One of the things you'll want to notice on this picture is you have the lens here deeper in the eye and the cornea is out over the outside surface. That is commonly mixed up. We have a couple different types of cells in the retina. The rods are the cells in the retina for black, white, and grayscale vision. These are able to work in dim light and also give us visual acuity. These are good at detecting shapes and movements. These tend to be more concentrated around the periphery of the retina, so we can see things coming up out of the corner of the eye. The cones are the cells able to see color vision. We have 3 types of cones that are able to see blue, red, and yellow type of colors. They're actually referred to as magenta, cyan, and yellow-green. When we see the colors, it is a combination of the cones being stimulated. The cones are most concentrated in the center of the retina. There is a small depression in the center where the cones are most concentrated called the fovea centralis. There is also a spot where the nerve cells of cranial nerve 2 extend into the retina called the optic disc. Where these cells come in, there are not a lot of retina cells, so this forms a blind spot. One of the links available to you, called Neuroscience for Kids, has instructions on how you can find your own blind spot. The vitreous body is a clear, jelly-like substance inside the inner eye. You may have seen this at the fish counter if someone punctured the eyeball of the fish. Human eyes will have the same substance inside. When light travels from one medium or substance to another, it will bend. We call this bending of light refraction. This is why if you open your eyes in a swimming pool and look at your hand, it appears larger than normal. Accommodation is the increase in the curve of the lens for near vision. By changing the shape of the lens, we are able to change the light it focuses, or change the place that it focuses the light. This works similar to mirrors you may have seen in an amusement park, which were bent to make you appear taller and thinner or shorter and wider. Convergence is the automatic movement of the eyeballs towards the midline or center. You can see this if you hold your pencil straight out in front of you and pull it towards your nose. Both eyes will follow it and move towards the midline. Here we've got a little animation of focusing the eye. So we can see the light rays coming in towards the eye here. You'll notice as the light moves into a different substance, that's when you're going to have the rays bend. The visual pathway is the path the nerves follow to carry the information from the retina back to the visual area of the occipital lobe. There are several drawings of the visual pathway. There will also be one that I've drawn that I feel is easy to use, which I will scan and put into a document associated with the nervous system. To make this easier, forget about your ideas what you've been told about what the right eye sees and the left eye sees. The visual information collected with the retina of the eye is going to be different than the visual information that is going to be perceived back in the brain. The retina is divided into a medial and lateral portion. The medial portion of the retina perceives the visual information for the lateral visual field of the same eye. The lateral portion of the retina perceives the visual information for the opposite side's eye's medial visual field. The retina then connects out the posterior side to the optic nerve. The medial fibers of the optic nerve cross or decussate at the optic chiasm and join the opposite side's lateral fibers. The optic chiasm is located by the pituitary gland behind the eyes. The medial and lateral fibers of the optic nerve join to form the optic tract. It is known as the optic tract after the optic chiasm. The fibers of the optic tract travel on the same side to pass through the thalamus on that side of the brain. After passing through the thalamus, the fibers change names and become the optic radiations until they reach the occipital lobe. You will want to print out this picture of the visual pathway handout and go through it a few times. To determine the effects on the visual field and which part of the pathway is damaged, all you need to do is follow the lines out to the visual field. An example of this will also be provided as a scan document. This is an additional picture showing the optic pathway in here. You can see where the light comes in and where they cross at the optic chiasm. The ear is divided into three regions: the outer ear, the middle ear, and the inner ear. The outer ear collects sound waves. It is made of the auricle, which is the structure on the side of the head and is skin over cartilage. The EAM, or external auditory meatus, is the hole in the side which connects from the auricle to the eardrum or tympanic membrane. Inside this canal you will find cerumen or earwax. The tympanic membrane is a thin membrane, commonly known as the eardrum, which vibrates when sound waves enter the ear. The middle ear contains the auditory or eustachian tube. This tube connects from the throat to the middle ear. This allows pressure from inside the middle ear to balance with the outside pressure by taking in air through the mouth and throat. The ossicles are the bones in the ears. These are very tiny and can fit on the tip of a finger. These bones include the malleus, incus, and stapes. They will transfer the vibration from the tympanic membrane to the oval window by hitting each other like dominoes. The oval window is the opening in the partition between the middle and inner ear. The inner ear contains a bony labyrinth which forms several of the structures of the inner ear. It will form the cochlea, semicircular canals, and vestibules. These contain the structures for perceiving sound and balance. The membranous labyrinth is a series of sacs and tubes inside the bony labyrinth. Endolymph is the fluid inside the membranous labyrinth, and perilymph is the fluid between the bony labyrinth and membranous labyrinth. The endolymph and perilymph fluid is continuous with the cerebrospinal fluid. It just changes names depending on which location it is in. The vestibule is an oval-shaped opening in the middle of the bony labyrinth, which contains the utricle and saccule. Static equilibrium tells you what position your head is in when it is not moving. This is detected by the utricle and saccule. One of these sacs is oriented vertically and the other horizontally, and they are lined with sensory cells called hair cells along the otolithic membrane. Inside the sacs we have small stones called otoliths made of calcium carbonate, which float on the surface of the hair cells. When the head changes position, it pulls the stones down to the lowest point, and we determine the position of our head by the particular hair cells that are stimulated. Dynamic equilibrium is perceived in the semicircular canals. These three canals run in the three planes of the X, Y, and Z axis, or vertically, horizontal, and transverse. They are also lined with sensory hair cells, which will perceive the movement of the gelatinous fluid inside, called the cupula. This will tell you if your head is moving by the sloshing of the cupula within the canals. The fluid is thick and gelatinous, so it will sometimes be a little slow to drip back down when it has been forced to the side by spinning. This gives us a feeling of dizziness when we stop moving and the fluid continues to slowly move down. The cochlea is the snail shell-shaped spiral canal in bone, which is lined with the organ of Corti. The organ of Corti is the actual sensory organ of hearing. It contains ciliated receptor cells on a membrane inside the cochlea. As the sound waves enter the cochlea, they will trigger a particular pattern of the hair cells to give us specific sounds we hear. Each sound wave has a particular size and frequency, which allows it to stimulate just certain hair cells like notes on a keyboard. Because the sound waves are a form of energy, it cannot just disappear into nothing, but must move somewhere. The round window is an opening that allows the sound waves to exit, where they will be absorbed into the mass that makes up the skull and head. Here we've got a short animation of the ear. This activity will identify the anatomical parts of the ear and learn the purposes and functions of these parts. So here you can see the three regions, the outer, the middle, and inner ear. So you will be able to go through and complete this activity with the practice test on your own, with either linking through the Integrity presentation or on Angel. This is the pathway sound waves will follow as they enter the ear. First, they pass through the EAM and vibrate the tympanic membrane. The malleus, incus, and stapes transfer the vibration through the oval window and cause the perilymph to vibrate. This vibration enters the cochlear duct and triggers specific receptors along the basilar membrane of the organ of Corti, while excess energy from the sound waves exits the round window. Here we have another animation on hearing. For humans, speech, music, and other environmental sounds form the basic elements of language, social relations, and adaptive response to environmental stimuli. Our auditory systems can detect rapid changes of sound intensity, measured in decibels, and frequency measured in hertz. In humans, the sound intensity and frequency roughly correspond to loudness and pitch. The task of the auditory system is to convert the changes in pressure in the environment into the neural activity that permits our brain to attach meaning to the sounds that we hear. Each part of the ear performs a specific function in hearing. The external ear captures, focuses, and filters sound. The middle ear concentrates sound energies. Finally, the inner ear transduces mechanical energy into neural activity.
The ear is responsible for translating variations in air pressure, whether from speech, music, or other sources, into the neural activity necessary for our perception and interpretation of sound. The ear can be divided into three principal sections: the outer ear, the middle ear, and the inner ear. Each of these parts performs a specific function in processing sound information. Sound waves are first collected by the outer ear, which consists of the external ear, or pinna, and a canal that leads to the eardrum. The configuration of the external ear amplifies sound, particularly at the frequency ranges of 2,000 to 5,000 hertz. Not coincidentally, most human speech sounds are distributed in the frequency range around 3,000 hertz. From the ear canal, the sound waves vibrate the eardrum, which in turn vibrates three tiny bones in the middle ear: the malleus, incus, and stapes. The stapes vibrates a small membrane at the base of the cochlea, the oval window, which transmits amplified vibrational energy to the fluids of the cochlea. The round window serves to dampen the movement of fluid within the cochlea. The complex structures of the inner ear convert sound into neural activity. In mammals, the auditory portion of the inner ear is a coiled structure called the cochlea. The region nearest the oval window membrane is the base of the spiral. The other end, or top, is referred to as the apex. Along the length of the cochlea are three parallel canals: the tympanic canal, the vestibular canal, and the middle canal. The principal elements for converting sounds into neural activity are found on the basilar membrane, a flexible structure that separates the tympanic canal from the middle canal. Let's take a closer look at the basilar membrane by unrolling the cochlea and peering inside. The basilar membrane is about 5 times wider at the apex of the cochlea than at the base, even though the cochlea itself narrows toward its apex. It vibrates in response to sound transmitted to the fluid-filled cochlea by deflections of the oval window initiated by the bones of the middle ear. High-frequency sounds displace the narrow, stiff base of the basilar membrane more than they displace the wider, more flexible apex. Mid-frequency sound maximally displace the middle of the basilar membrane. Finally, lower frequency sounds maximally displace the apex. Within the middle canal and atop the basilar membrane is the organ of Corti, the collective term for all the elements involved in the transduction of sounds. The organ of Corti includes 3 main structures: the sensory cells, or hair cells, an elaborate framework of supporting cells, and the terminations of the auditory nerve fibers. The heights of the stereocilia increase progressively across the hair cell, so the tops approximate an inclined plane. Atop the organ of Corti is the tectorial membrane. The stereocilia of the outer hair cells extend into the indentations in the bottom of the tectorial membrane. The movements of fluid in the cochlea produce vibrations of the basilar membrane. These vibrations bend the stereocilia inserted into the tectorial membrane. Depending on the direction of the bend, the hair cells will either increase or decrease the firing rate of auditory nerve fibers.
So in this tutorial, we have seen how the ear captures sound energy and transduces this mechanical energy into nerve impulses. The external ear captures, focuses, and filters sound. The bones or ossicles of the inner ear focus the sound energy into the oval window, generating pressure waves that sweep along the basilar membrane of the cochlea. The structures of the basilar membrane allow it to respond to vibration of different frequencies. The organ of Corti contains the sensory hair cells, about 15,000 of them, in humans, that convert the mechanical energy into action potentials that are transmitted to the cerebral cortex. You will also be able to access this on your own so that you can complete the practice quiz to test your knowledge. Cranial nerve 8 uses the cochlear portion to carry the information from the organ of Corti to the lateral temporal lobe to experience hearing in the brain. The vestibular information is carried to the medulla oblongata, where it is coordinated with information from visual input and mechanoreceptors. Motion sickness occurs when this information does not agree. There is a great variation in the sensitivity of people to the disagreement. Some people actually enjoy the feeling and seek it out on amusement rides, where others will be absolutely miserable with motion sickness.